All right, everyone. So we are moving on to chapter 10 today. This is going to be talking about genetic engineering and literally making use of everything that we've just learned about genetics in the previous chapter. We're going to make use of all of that information and put it to use for our own good. So medicine and manufacturing proteins and things like that for our benefit, or to study diseases so that we can better, make medicines against them or vaccines or anything like that. Now, genetic engineering. Okay, so let's start off with restriction endonucleases. These are enzymes. So enzymes are anything that help catalyze a reaction, organic molecules that catalyze reactions, right? Lower the activation energy and help us get from A to B faster, more effectively. So these enzymes are naturally derived from bacteria. Usually they're used as a defense mechanism to get rid of foreign, nucleic acids like plasmids they don't want or something like that. But now we're going to use these guys to our advantage. And essentially these enzymes cut DNA at palindromic sequences, so the same forward and backwards. And they leave little sticky ends, and we can use that information that we know about how those specific restriction endonucleases work and hijack that information for us to be able to insert our own genes into plasmids or something like that we can put into bacteria and then force bacteria to make whatever protein we want them to make that way. So essentially, if you have a plasmid, essentially, if you have a plasmid that looks like this, and it has a site here that we can cut with a restriction endonuclease. So we'll have, remember, it is double-stranded. So I'm just going to kind of show it like this. It will cut it and leave these sticky ends and so on and so forth. This would be a circle, but you get the idea. So the sticky ends. Originally, these guys matched up here. Okay. This is a terrible drawing. I'll make this clear and it'll be worth it. Okay, so this is the whole entire thing of the entire circular plasmid. Imagine that that's going on and on for the circle. Okay, same on this side. We clip it at that site right there with the restriction endonuclease, whatever it is. It cuts at that palindromic sequence. And then it leaves our overhanging sticky ends. This is our overhanging sticky end. See how it's just one strand and it's got a little end hanging off. And this is an overhanging single stranded sticky end. And these guys, usually like maybe 10 base pairs or something, this little area here. So this area would have bound up with this area. So now it's open, but we can stick our own DNA that we know has that same overhang on it. So the one that'll match up with the top one and the one that'll match up with the bottom one on their little overhanging guys, and then just stick that whole bit into that slot. And this codes for, I don't know, insulin or something. Okay, so that's the idea of how that would work, using restriction endonucleases to cut at specific places, known places in DNA, and then we can use that to insert known genes into those spots, and we can sequence it to confirm it and everything like that. So it's not all guesswork or anything. We can actually confirm everything. One of the more useful tools that we can use in genetic engineering would be PCR. That's polymerase chain reaction. We can take a tiny little bit of DNA and amplify it out into exponential amounts in short time. You can start with your double helix. This is a double helix. Let's just pretend like that is a big, long, old double helix for, I don't know, a gene that helps you identify E. coli. And you swab an area at a food manufacturing plant to see if E. coli is found in that particular area or on a food sample or whatever. And now you're going to pop it into a PCR. You have primers that look for a specific E. coli gene that helps you identify it. And what happens is heat. So that's what this triangle represents. It's a really good triangle, by the way. Heat will add heat, and then that will split apart the two strands. Then we'll lower the temperature a bit, and we will stick on some little primers that we have already manufactured and made because we know what gene we're looking for. And then we will add a polymerase that is heat stable, because remember, we just had to heat things up. So it can't be something an enzyme that is easily broken down by heat. So heat stable DNA polymerase from Thermus aquaticus bacteria in the deep sea vents that exist in the like boiling deep sea vent area. That's what we call it TAC, T for Thermus and AQ for aquaticus. And anyways, that's how we use that. Put a little primers for our known thing, and then we use the polymerase, and then it makes a copy, and now we have two. And then after that, you will have two, so that'll be four. And then, and so on and so forth. It would be doubling essentially every single time that we go through a round. And so it doesn't take very long to get through a round. And so we go through that round, many times, hundreds of times. It only takes a couple of hours to get up to billions of copies. So it's pretty cool how that can work from a very, very small amount of a DNA sample. You do have to, it only amplifies a known target. So it's not like you just, somebody's sick and you want to know what they have. It's you swab and then throw it in a PCR machine. You have to know what you're looking for. Are you looking for strep, or are you looking for diphtheria? Something like that. So you have to know what you're looking for, and you have to have primers available for that thing that you're sequencing for. Otherwise, the PCR doesn't work. So you have to have the primers, and you have to know what you're looking for. Not just rando, whimsically test for whatever you want. Okay, so we went through the steps a little bit. Heating denatures or separates the strands. Denaturation via heat is something that we can do to proteins as well, but in this case, we're just talking about nucleic acids. Okay? So separating our strands using primers that allow us to build off of something. You have to build off something. We talked about that in the previous chapter. So there's little sequences that the polymerase will build off of. Those anneal or stick onto those separated strands when we lower the temperature a little bit. Then the DNA polymerase, the TAC polymerase, will add matching nucleotides, A to T, and then G to C, etc. And then it will just keep doing that over and over and over again. Denature, anneal the primers, and then elongate, and then go over and over and over. DNA can be introduced into target host organisms via plasmids, which we had talked about with that restriction in nuclease, right? You pop your new gene into a plasmid, pop that plasmid into a competent bacterial cell, right? So we can use plasmids for that. We can even engineer viruses or bacteriophages to carry those genes for us. And we can even use artificial chromosomes that have been engineered like down to detail for maybe even several genes that we can insert into bacteria so that they'll produce those products that we want. GMO is a genetically modified organism. These are transgenic organisms, and these are organisms that have cloned genes from other organisms put into them to enhance them somehow. Whether that make them resistant to certain antibiotics, or whether that make them producing, products that we would be interested in, like insulin, when we do with bacteria. Those are all going to be GMO and transgenic organisms. We are not altering any other part of the organism whenever we do that, so do keep that in mind as well. Gene therapy is a term that refers to hijacking viral latency to insert functional genes in humans. Basically, you can take something like HIV. HIV has the ability to put its genome into your genome, into your cells, and insert its DNA into your DNA. Well, if we replace HIV's genes with something that a person can't code for naturally because they have a mutation, they aren't able to make a certain enzyme like insulin or something like that, we can, engineer the HIV to carry those genes instead of its own genes, insert those new good working genes into that person who is sick and can't make that protein. And hopefully that gets incorporated permanently into their entire system. And so now they can make whatever protein was missing, whatever. This is still something that is being developed and hasn't been officially approved through FDA use yet. There are a lot of labs looking into making this work as best as possible, but make it as safe as possible as well for patients to use. But this is just definitely probably one of the hottest topics in genetic engineering and medicine nowadays. If you've ever heard of CRISPR or people using CRISPR to put genes into their own cells, That's what this is. That's what gene therapy is talking about, but without having to use the virus as the intermediate. So anyways, that is that for genetic engineering. And I will see you guys for, I feel like chapter 24 and 25 are more boring, but I'll see you guys for chapter 24 and 25.